Welcome to PMTV AP Class 3 Bio Review with me, Andrew Dufresne, Senior Tutor at Prep Matters. Thank you. Thank you for joining me in my very nondescript corner here in Washington, D.C. Let's jump right in. And today's class is going to be about genetics and inheritance. As you can see, we've got another few classes planned to amp this up towards the magnification level to bring it up to the cell, and then check that out. As an entity, right now we've been dealing with all of those very, very tiny bits inside the cell. So if you're not familiar, I just want to share that if you have a question, there's a raise hand feature, and then also a Q&A location to ask your questions. I'll see those and then get to those as best I can as I go through. You can also send questions to classes@prepmatters.com. All right, so just to remind you about why we are where we are with the level and magnification that we're talking about for the cell and AP biology. We're connecting a number of different topics here. So we have the concept of evolution or change and adaptation to your environment over between generations. Energetics, this idea that the energy to do this stuff has to come from somewhere. We talked about a little bit of that in terms of the reactions and everything. Always trying to shoot for the easiest situation in life. Information storage and transmission. That was the big topic last week with nucleic acids, which is where we're going to really pick up and then get into how these different systems are interacting as well, all the while keeping an eye on the kinds of skills and tactics the AP Bio test is expecting of us. Think like a scientist, simply observe, come up with some kind of description of what may be happening, and then how would you test that, and what would go into that plan, and then what would you get for results, and how would you analyze those? We're going to actually talk about that when we go over Mendelian Genetics. So, so far, again, to incorporate those big ideas and bring in some other thematic elements that I really think are going to set the difference between when you're prepping and preparing for this exam on your own, and kind of looking behind the curtain and seeing what is it that I want to be keeping in mind here. Well, how are they going to formulate the questions on this very bizarre pandemic edition AP exam? So we talked about smaller things coming together. Monomers ended up forming polymers. Nucleic acids ended up getting turned into proteins. But even that was really just a lot of different chemical molecules coming together with their own properties to form a larger, more functional structure. We talked about that relation between structure and function. Basically, if you have this kind of nature to you, then you are probably going to naturally be inclined to do this action. And how can I take advantage of that? How can that basically perpetuate processes that I want in life? And then, that sense of not directing it towards a particular goal, but just staying in line with random things happen, and the thing that works sticks around. All of that stuff that we talked about with nucleic acids in the beginning of the world, the RNA world hypothesis, it's all within that context of anything could happen, and this is just simply what we had reason to think did happen, and it works with everything that we see. I already kind of mentioned previously that idea of the lowest energy and whatever simplest, shooting for that. That's a constant in the universe, whether you're a teenager or literally just the randomness of the universe itself. Okay, and then lastly, and this is going to be big in today's lesson, which is why I've got this at the end here, inputs and outputs. So a lot of the things we need to know for AP Bio, a lot of the information and the knowledge is cycles or systems or series of steps. And sometimes there's a lot of detail to each of those. That can get super distracting or daunting. You don't want to worry about that. Focus on what's going in and what's coming out. And if we include all these other kind of thematic elements and the big ideas, then we'll naturally come to, this makes sense. This is the logical progression. This is what I need to focus on. And I'll always try to mention things that will be beyond the pale for this test. We're going to talk more about the other information that College Board has let out in terms of how you're going to be assessed. But if you have any questions, Q&A or classes@prepmatters.com. I know a colleague of mine shared his social media handle. I'm at Mental Markets, MENTALMARKETS. You can also tweet me there. And if you hashtag PMTV, then I'll see it and be able to answer that as well. All right. So I wanted to just cover a little bit more in terms of the content itself. That was a lot of themes and things you want to keep in mind as you're going through this. But remember that elements and their properties are going to come together to make molecules, which then have their own properties. Those came together to then actually lead to something that could encode for something else, like nucleic acids. This is going to come up when we talk about the idea of genes and alleles and traits that may be polygenic. If any of these words are not familiar to you, you're going to want to be familiar with them, but I will discuss them more when we get to that. Also directionality and endedness. This is also going to be big as we're going to talk about meiosis. We're not going to get too much into cell structure or anything like that. I want to leave that till the beginning of next week. I'm just trying to bridge out from, okay, here's the nucleus, nucleic acids, chromosomes. I mentioned those aspects last week. Basically, how does that get passed on? So we're going to talk about a couple different ways to approach that, whether it be systematically, like functionally, how does that happen, meiosis, and then what does that even mean? Why do I care? How does that division, how does that passing on of genetic information, what does that translate to? How do we see that? And that's going to be the genetics and Punnett squares that I'm sure you guys are expecting. Okay. So a couple of things that I didn't actually get to last week that I want to mention this week because I think that they are a good marriage of the two topics are mutations and biotechnology. So in order to understand mutations, we have to know a little bit more about RNA and how it encodes for amino acids. And then after that, we are going to go from the biotechnology and how we kind of figured all this stuff out in the real world, rather than just being information that you're expected to learn and never question. And also, how do we add more information to it? Well, if we know the tools, then we can do that. We're going to go from there and then talk about the chromosomes as our fundamental player, right? So we're going up in those levels of magnification. There's going to be a little bit of mitosis simply to give you a framework for meiosis. There's some similarities. You may have it in your head what you think those are. Maybe we're aligning in how we're seeing it and thinking about it. But I think that again, if we see those patterns, you realize this is less for me to kind of just memorize and more for me to kind of logically understand in one way, and then I can apply that understanding a number of times on different topics. Okay, and then, like I said, what does this matter? How do we see this? That's going to be the Mendelian genetics and then the other forms of inheritance. Any questions so far? Any questions from last week? Anything else going on here? Okay. So, triplet code. Number one question I usually get, oh my God, do I have to know that? No, you do not have to know this. You'll have to know it perhaps a little bit more if you study biochemistry in college. If you got some spare time, the spare pandemic time, sure, give it a memorization and see how useful that is. It won't help you on the AP Bio test. As a reminder, what the heck this means and how this plays a role. DNA, A, T, G, C, not U, but T. That has a template and coding strand. And then the RNA polymerase comes in. Which does it sit on? Template strand, the coding strand. Do you remember? It's a smaller question, not too worried about responses. I want to keep going, but I want to give you that second there to think about that. It sits on the template strand, and then what it produces is complementary to those bases, and that resembles the coding strand. Template is what we work off of. So we get that RNA strand, and now we have uracils instead of thymines. And then there's this whole idea of we have to all of a sudden make an amino acid chain using ribosomes and tRNAs. I mentioned this stuff last week. Yes. Now, how does that actually work? Well, we observed the amino acids that actually existed. And then we align that up in terms of how those are produced according to what we had for a sequence. And we did a little bit of math, this idea of like, well, could it be 2 amino acids? I'm sorry, 2 bases for an amino acid? What if it's 4? Turns out it's 3, and it's simply because that's the lowest number, where 4 different letters, 3 in a row, provide enough combinations. Sorry, permutations, in the event you are also doing pre-calculus right now, or any kind of AP calculus class, because the order does matter. So the number of permutations that we needed to get the number of amino acids, we have a triplet code. So essentially just keep that in mind. We have a particular first, second, and third base in order that then translates literally to a particular amino acid. You don't need to know all the amino acids either. And then that is how we make our protein. What could go wrong? Well, many things. So nucleic acid, we talked about stability differences with DNA and RNA. RNA single-stranded, DNA double-stranded. DNA is a bit more stable, but mistakes still happen. In fact, they are a big component to evolution and natural selection. You won't be needing that unit on the test, but I dare say that AP Bio has it in their interest to have you be thinking about that concept, that big idea throughout these units. So that damage that can happen to the DNA can change things up, and if that change is beneficial, then it can stick around. And that's actually how we get change throughout generations. There is no evolution within the generation. That's not a thing. So mistakes can happen, and while we want to correct them as often as we can, because we at least know the way that it's written works, we're alive, and we're functioning. So, hey, let's keep this rule book, this recipe book, the same. But we need to have a way in which to fix that. And so there are enzymes that come through and they correct bases, and sometimes they even make a mistake. So suffice it to say that if this happens and it's an area of DNA that's never expressed, then it doesn't really matter. If it's an area of DNA that is a big deal gene, then it can have really big effects and everything in between. So what kind of mutations do we want to be aware of? And this is mostly just that you know these words, you don't freak out, you see them in a question and you mosey on and you have a good enough idea about what the heck they're talking about. Point mutations versus gene rearrangements. Point mutations come in 3 forms: nonsense, missense, and silent. So I think the way to remember these, and how I often tell my students, is if I were to be speaking through a slide and then, what did I say? I didn't say something wrong. What I said was just nonsense because I just stopped abruptly. So if that happens, if you get a change, you can go back in the video or open up another tab or something with the triplet code if you want to see it. But there are a few codes that are for stop codons. That tells the enzyme you're done. This message is over. But if you put that on too early, you don't get the protein that you expected. That's a nonsense mutation. Missense is when you change the amino acid. So you made a mistake. So if I put a word place wrong, I think I still was missing a preposition in that. So it's difficult, based on my editing mechanisms in my body, to make a mistake of that nature on purpose. But if we put a different amino acid in the spot, then that can change the properties, that structure and function connection All of a sudden, the R group of an amino acid is different, and now that area is different. We have different alpha helices, different beta-pleated sheets. The function of the protein can be different. Now, we also mentioned the whole math of how many bases we look at, 3, in order to get every amino acid. It's a degenerate code. There are a bunch of ways in which, I'm not throwing shade at it, this is what it's called. There are a bunch of codes that are for the same amino acid. If I were to say a word in 2 different pronunciations, penalize and penalize, they're both acceptable. That would be a silent mutation, one where you're changing it to something that basically means the same thing. It's another amino acid of the same, it's another code for the same amino acid. And then there are gene rearrangements, and these are going to come up in the genetics bit as well, which is why I think it's a good foray, good segue rather. Frame shift, they're basically a insertion or deletion. Because it's a triplet code and it's in order, if all of a sudden you were writing a 3-letter word ant, A-N-T, and then you just put in an extra word between the A and the N, you wouldn't have that word anymore. So when we insert segments of genes, then it's or delete them, cut them out, then what is left behind is a frame shift. It no longer was the length that we expected, and you can truncate proteins, also the stop codon is closer, or you can just completely obliterate its function by inserting something that doesn't make any sense. Duplications, I think, self-explanatory. Basically, you can have double the expression of something, but you can also end up having that new thing no longer need to do that function because the other copy is satisfying it. And so it can go on and do something else, and now it's a different ballgame. Inversions basically flip the script. All of a sudden, that directionality that we talked about that's so important, that gets messed up. And that's going to change what we're going to get out of that. If that's the input, the output would be different. Translocations are the movement of segments from one chromosome to another. We talked about how the DNA and proteins condense down and form those characteristic X's of chromosomes. If we take parts and tips of those and stick them onto other ones, then that doesn't mean that, oh, well, the genetic code is there, what does it matter? No, there's all these other elements that determine how much of this I want, or how little of this I want, or which cell this goes to and gets expressed in. So when we have translocations, it's very bad news. This is actually at the heart of a number of kinds of cancer. And then transposons. They're actually designed to be, they excise themselves out of the genome and then they pop over and go somewhere else. It's kind of like a frame shift situation, except that they themselves are gene segments that are bouncing around in different spots. It's not just, oh, we're going to make it a problem here, give you a problem in multiple different locations depending on the cell or chromosome. Okay, any general questions about that? Checking on the time here, I think we're doing okay. So less on the slides, more on the explanation. There's a lot here, boiled it down. So let's carry on. Okay, biotechnology, this idea of how do we know what we know? We have found ways to bring together segments of DNA that we wouldn't otherwise see, and that's recombinant DNA. And this allows us to then put genes into other things and use those as tools or get something else out. Everything from the production of certain proteins in the milk of cows, with transgenic cows putting in new genes, or a really common lab tool would be green fluorescent protein, something that naturally occurs. That bit naturally occurs in some fish, but we can take that gene out, put it in something else, and now that cell will glow green. And so that can be helpful as a tool. Plasmids, that circular bit of DNA as opposed to our chromosomes, that's the easiest stuff to move around. Bacteria, hello, welcome to the party. This is going to be how we're going to use you from here on out. Vectors are basically just plasmids that have a few more extra things in them. That could be something like antibiotic resistance or a site for restriction enzymes. You can cut the size that you want. Again, I know I'm expecting some background knowledge here. This is review of the big things on each topic. Transfections, that's the transformations first, bacteria, transfections, viruses. We don't need to go into too many details on that. Again, these are just some words that you may come across in the free response questions. So I want you to at least look into them well enough in your studies so that you aren't spending some of your precious time during the kind online situation that we're going to have looking up what that word means. PCR and gel electrophoresis. Okay, so perhaps a couple of the most common things in a crime show on TV, and PCR is so basic in the lab, I laugh every time someone makes a big deal out of it. But it's actually a really important tool. Polymerase chain reaction. It allows us to make a ton of copies of whatever segment of DNA or RNA that we want. And we change the temperatures, we throw in all the ingredients, and we change the temperatures so that we basically make it go through the process again and again and again and again and again. And when it does that, it produces more and more copies of what we want. And then it's easier to study. Gel electrophoresis is when we run a current through, think of it like Jello, where it's powder and water, you got to mix it together, and then you have pores, areas where there wasn't powder or there's air or something like that. Different size samples, in this case nucleic acids, run in different lengths based on the current running through it because it has charge. We talked about that last week as well. And then also its size, its ability to get through the pores in the gel. I don't want to go too much further into RFLPs, DNA fingerprinting or sequencing. Again, just the idea that be aware of what those are and what is used for those. Something like restriction fragment, like polymorphisms, involve restriction enzymes and how they cut, and then the different lengths that you would get, but how each of those are unique to each individual. Okay, now we're going to be getting into all this new stuff. And I imagine, I don't see any questions here at the moment. This is great. It's a lot of vocabulary for you at this time. I wanted to kind of, create my own little presentation version and slide rearrangement and put the vocab up front, simply because it wasn't about the definitions. The terms are becoming more and more niche and specialized to the topic. I think the earlier terms are really important because they're the foundational ones. They're the ones that all these other ones often have a more basic version of themselves that you can hark back to in your understanding of these more higher order complex processes. Okay, so ploidy, what the heck is that? And why doesn't it have a red squiggly line like it's misspelled? Because it's a word. It is the number of chromosomes in our genome. Remember, all of our genes, right, together make a genome. So what do I mean by that? Well, it's not the number of chromosomes in terms of what the species has. It's about the copies that you have in your cell. That means haploid or diploid. We're just going to focus on those for us. Are there others? Yes. Tetraploid, octoploid, twos. It'll make sense when we go through the math of meiosis. There is not much. But these different ploidies in other species, it doesn't really pertain to how we're going to be approaching this stuff now. Just be aware, again, that fruits and plants, they're crazy. They've been around for a long time. And spring is amongst us, so they are getting together. My eyes know it from all the pollen. And there are many versions that are more than diploid. So they have more copies of their chromosomes from parents than we do. Haploid, half, diploid, di, 2. What does that mean? N is the letter you typically see with haploid, 2N with diploid. Our haploid number is 23. Our diploid number is 46. Every cell in my body that is not producing a germ cells are sperm and egg, your gametes, all the terms on the left here, in the first 3 bullets, haploid, germ cell, gamete. These are all my N, half the genetic content. All the content's there. We'll get into slight variations on that, but it's not a full set. It'd be like having every card in the deck, but not all 4 of each one. So in this case, diploid is 46. That's because we have 23 chromosomes from each parent, mother, and father. Germ cells, I mentioned, sperm and egg, and those that go on to form gametes, which get together and form zygotes. Details out because they are the details that will be different in meiosis. All right, we've got a question. What happens to the other organelles during mitosis? This is a great question and something that we will explore more when we come back to mitosis. Everything gets doubled. The organelles, like, you don't get all of a sudden, oh, I got all the treasure and you got nothing, or oh, I got the mitochondria, you didn't. There already is more than one of everything. We have this idea that, oh, there's the mitochondria, it's the powerhouse of the cell. It's not just, a mitochondrion and then mitochondria, plural. We have multiple of them in our cells. The same way we do ribosomes, and I mean, the endoplasmic reticulum and Golgi apparatus, these do get duplicated for different cells, but again, essentially both cells, like in interphase, you have all this other kind of stuff happening. The replication of DNA, the checking for mutations and stuff like that that we mentioned earlier. The increasing in size so that you don't end up with 2 tiny daughter cells. So all of that is taken care of that way. Thank you for the question. Okay, so I told you the details we're going to keep in mind. Let's see how well you're doing that. Whoa, this looks more complicated. Turns out it is. So meiosis, let's bring it back to the initial question that we had. How are we passing on this genetic information from generation to generation? We kept talking about all of this diversity that can come out of this from the different combinations of amino acids and different chemical interactions and proteins you can make and all this stuff. And then we also talked about how mutations introduce this new element that can lead to more diversity and change that can then be beneficial for society, survival. So this is good. It's good that it's complicated. This is a part of sexual reproduction, a just mathematical advantage to diversification in terms of your gene pool, in terms of the traits and all these things that we're going to talk about more after this functional take on inheritance. It increases or broadens and deepens the gene pool, and then greater diversity, more likely that we are going to survive as a species, because it really does come down to the collective, despite how it does not feel that way many times. We're like, I can't worry about the rest of mankind. I've got this one test in May, the AP Bio exam. But that diversity is great. We bringing into current news the SARS-CoV-2, the coronavirus. Some people are asymptomatic carriers, they never get sick. Some are symptomatic and it's mild, and then some are severe. We would all respond the same way were it not for diversity. Our immune systems are very different, and that actually is an example that brings in this idea of environment along with our genes and nucleic acids and their specifics. And we'll mention that at the end as well. So back to meiosis. It's very complicated. So let's not focus on all of this on one slide. Do you see how it's still prophase, metaphase, anaphase, telophase in cytokinesis? That's why I brought up mitosis. But then it's that again, and we want to figure out why that is. So let's take the first one. Prophase 1 is prophase, metaphase, anaphase, and telophase 1 is pretty different than mitosis. Oftentimes we can think that it's more like mitosis because we're like, oh, it's just going through the same process, inputs, outputs. We start diploid. We're making gametes. These are the germ cells that are making gametes, but they themselves have all of the information. So we start diploid, but those daughter cells at the end, they're haploid. already that is very, very different from mitosis. And that is definitely something you're going to want to know. Meiosis 1 is where you go from diploid to haploid, where you go from a copy from both your parents to not a copy from each of your parents and separate daughter cells. Oh no, it's more complex than that because you have this thing called recombination. And this adds even more to the diversity win from sexual reproduction. We're not just satisfied with leaving chromosomes from our mother and our father as separate ones and that being diverse and jumbling up which ones of those we mix together. No, we also have crossing over, translocations, those gene rearrangements that we talked about before, but this is to produce gametes. They're not cells that are being expressed. They're not our body cells. This is a different kind of, it's a similar process, but it is different when it happens in meiosis and we're making gametes instead of somatic cells. So what do we have that we want to focus on here? Diploid to haploid, recombination, crossing over. Prophase still has that idea of condensation. We have our DNA replication as we typically would. And the nuclear membrane dissolves and breaks apart. We're having the centromeres, centrosomes, made up of centrioles. Again, the same things from mitosis. We don't have to worry about it too much. Ejecting these spindles, creating the highway for our chromosomes in the cell. But what's happening here is that we have homologous, since we're talking about mother and father here, we're breaking down the genetic content a little bit more. We have, we'll call them maternal and paternal chromosomes. Chromosome 1 through 22 and sex chromosome, and then 1 through from the mother, and then 1 through 22 in the sex chromosome from the father. Those are all duplicated, but then they also line up with each other. Chromosome 1, the pair, so the 2 red in this slide here would be one of them, 2 blue from another. Whereas this would be a different chromosome number, and then this smaller one down here would be a different chromosome number. By all means, speak up if you can't see my cursor or anything like that, and I will try to get around any kind of visual aid there. So what happens when they do that lineup, they can have crossing over. They can have the chromosome tips cross, entwine in some fashion, and then when they separate, hold on so tight that they basically exchange. And so when that happens, they then line up as they would in metaphase 1. Here, before we were just making sure one copy of the DNA was on either side so that when we pulled apart, we're identical. Now, we are lining up according to number, not in numerical order per se, but that metaphase plate, that midline, is in between the homologous pairs of a chromosome. So sister chromatids are the 2 copies of that chromosome, the copy, the replication. And then homologous chromosomes, chromosome 1 for mom, chromosome 1 for dad. Another word you may see, tetrad. Both pairs are together, and then we have 4 to tetrad. So the plate basically separates between not just mother and father copy now, because now we could have had crossing over, which means when we go to anaphase, you can see, I don't know why they didn't maintain that necessarily in the drawing in metaphase, but in anaphase, you can see how it's red, a little bit of blue, red, a little bit of blue, blue, a little bit of red, going to the north, and then the others to the south. Are they actually magnetically aligned? No, but top and bottom. So that adds more diversity again, because now you have chromosomes from mom and then other chromosomes from dad. So that's even more diversity. This is celebrated when you're trying to face unknown changes in the environment. Okay, so then when those separate to either side, telophase, cytokinesis, they separate. They're not identical, and they're also haploid. So then what's going on in meiosis 2? If meiosis 1 was the big show on the road, what's up with meiosis 2? Well, they're haploid, but I don't actually have, I had replication of my DNA. right? So what you want to determine here is get down to, I just want one copy of that version of that chromosome. So we were talking about before mixing chromosome 1 from mom, and 2 from dad, and 3 from mom, or whatever. But I only want one copy of mom's chromosome and one copy of dad's chromosome. This is why also in DNA replication doesn't change the ploidy of the cell. So these are starting off haploid because they were the daughter cells from meiosis 1, and they end haploid. The daughter cells from meiosis 2 are also haploid. They're not also finding more genes to then become diploid, more chromosomes. So this is actually more like mitosis. Sure, it's a haploid version, but it's essentially very, very similar. By all means, come to me with any questions you have on these processes, but I wanted to kind of just cover this as here are the basics you really need to know. We didn't get into some of the structures there and what their functions are and all that, because at this point, it's really getting into that process of input-output. Same thing with glycolysis and the Krebs cycle and electron transport chain, which we'll talk about on the energetics class. But when you're getting into these processes, we're on the next level here. So now instead of that structure and function and getting into the weeds there, we're talking, well, what's just the organization here and how does this, what's the logical meaning behind this? Why would this happen? What is this? What do I start with and what do I end with? Okay, so now I have 4 daughter cells from one meiotic event, 4 daughter cells that are haploid from my one diploid parent cell. And this happens in our germ cells, and these 4 haploid cells are our gametes. Okay. With all of that, and talking about gene rearrangements, and talking about the value of diversification, and being able to then face unknown things, what does that actually look like when we're not inside the cell and we're actually just looking at the organism itself and how this plays out? What we're kind of getting into that is with that perspective is genotype versus phenotype. Genotype is our set of genes, and phenotype is our set of physical characteristics, how they are expressed. Genotype does not always match phenotype. It's certainly influencing it, causing certain possibilities for phenotype. But there was a monk in the 19th century, and I don't know, maybe he either had some time on his hands or he just simply was as excited about biology as I am. But I want to mention him only because if you are doing this class and learning this information, you got to know Gregor Mendel. Mendel, I'm not entirely sure, probably Mendel. But it could have been other individuals, other scientists that had discovered this stuff at the same time. This is always an interesting aspect of science. We learn about certain individuals doing this, but maybe he just had the better publicist and better access to share these ideas. But Gregor Mendel in the 19th century liked his true breeding pea plants. What did true breeding mean? That when his tall pea plants had baby pea plants, they were also tall. And when those had baby pea plants, they were also tall. So you always got the same thing as the parent. And it's an interesting caveat to how his research went, because it led him down this road of these 3 laws. These are the ones you're going to want to be familiar with: dominance, segregation, and independent assortment. But it really was based on his working with true breeding pea plants. The law of dominance. Okay, now what I was hoping to do here was write in these boxes. And this is a little different right now. So we're going to do this as quickly as we can. I'm actually going to stop my share here. Hello. And then I'm going to change it to just be okay. That actually, well, okay. So let's change this up. We are actually going to do old school. Bear with me, this is thinking on our feet. to show you what each of these laws would mean. Okay, so law of dominance. What did Mendel determine? I have this handy way of drawing my own Punnett squares. Okay, so I can refocus that. Hopefully everyone can see well here. It's a little different, I know, but hey, I want to go through this with you guys and help you understand what is going on there. So, law of dominance. Essentially, he had tall plants and short plants. And remember, they were pure breeding. So tall plants beget tall plants, short plants beget short plants. So he realized when he put together a tall and a short plant, let's just go with the expected nomenclature of capital T, lowercase t for tall and short. Now, this is the best my handwriting is going to get, but also I know that I haven't talked about this idea of nomenclature and how we write all these. Again, I'm expecting that outside review. These are alleles. They are different copies of the same gene. This would be something like this came from this plant's mother, this came from the plant's father. And they got together to make this particular gene wherever they are located on a chromosome. So the law of dominance, back to that. When we do our Punnett square, what we end up seeing, or what Mendel ended up seeing, did he end up doing this right off the bat? No. What spurred him to look deeper was the idea that, oh, well, here I have big T, little t, every single one. All of these plants, they were tall. So despite mating a tall and a short, he got tall plants. All the law of dominance means is that one allele can mask the effects of another. That's what that means. And you want to be aware of that when you're making your own Punnett squares for your response questions. Let's keep it going. The next would be the law of segregation. Now, what he means there is this, back to this idea of alleles. If I were to then take these guys here and cross those pea plants, told you that was going to be the nicest my handwriting would get. What we see here, these again are genotypes, not phenotypes. That's the phenotype for all of these, what we call the F1. If these are the parents, this is the F1 or filial 1 generation. This would be F1 and F1, and then this would be F2. Again, just some terms I want you to be aware of when you see these in your questions. So how does this work? Well, now we have tall, 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 short. Well, that's weird. I had both tall again, but now I have a short plant. That short plant makes it clear that this is actually how this is working. Again, we're kind of coming at this from a sense of, I already know the answer, I'm explaining the answer, working off the answer key rather than the actual act of discovery. All of those other questions that may be about how this all came to be, they're great, send them my way, but they're not going to be on the AP Bio test. So this is the law of segregation. These alleles segregate independently. Not to be confused with an independent assortment, but the idea is that you can have a tall plant that has a tall allele and a short allele. Those will separate, and that means that it's possible, if that short allele gets together with another one, you can have a short plant despite both parents being tall. Now, lastly, the law of independent assortment. This is where Punnett squares just turn it to 11. Go ahead and make sure that you can see that. And if you're wondering if you can draw a box on a piece of graph paper and not actually hit any of the lines, the answer is yes. So, okay. Told you it turns it up to 11. I can't even estimate apparently with this. Tables in PowerPoint would be so much neater. But there's a thing called a dihybrid cross. Di, two, hybrid, a mix of a couple of things. What am I actually talking about? Well, sometimes you can end up having more than one trait get inherited with the other. And it's not actually. Sometimes it's a lot of the times. It's your chromosomes have multiple different genes on them. And so when we're looking at more than one trait in an organism, then all of a sudden now we're talking about, well, how are those alleles going to work out to be different combinations here, considering both genes. So what could we do here? Let's see, we'll stick with the tall. So we're going to say that these guys are the big T and little t. Now again, this can seem like, oh, but then all of a sudden you doubled the amount of ploidy or something like that. No, no, this is just for the combination. While I think about another letter to put here, that is easy to tell, capital and lowercase. I've chosen H, even though we're talking height here. But now you see all I'm actually showing in a dihybrid cross is how you can have a capital dominant, lowercase recessive. Dominant alleles are simply the ones that mask recessive. See, I'm just including nomenclature as we go along. But a dihybrid cross now can now have all these 16 possibilities, because one allele, tall, and then maybe hanging over versus a crawler on the ground. I am making up this trait on the spot. But when these get together, it can be either allele if we actually see the next generation present kind of traits in this way. So Mendel saw these different phenotypes, and I'm not going to fill out the whole thing, but you've got this, you've got this, every possibility that you could think of is here. They're all accounted for. And what that means is that we have many different ways in which these phenotypes could be presented if each genotype made a different phenotype. That's often not the case because of the law of dominance. But what Mendel determined is that genes also, or traits, they also segregate independently. So just as the alleles do, so do chromosomes. Okay, so that was what I wanted to get through on those. Do you have any questions about any of these Punnett squares? I know we're rubbing against the end of the time here. Just a little bit more to cover, and then we're gonna have that practice question. Okay, so let's go back to me. Hello again. And then let's go to this. So there are other ways that things can be inherited. And you want to be aware of those as well. Here we go. So what am I talking about? Well, you're going to see things like pedigrees, potentially. You could see, I know it's just a couple of questions, but you could see a pedigree. And that is those squares and circles, males and females, how those come together and everything. You could then be asked on that pedigree to talk about sex-linked genes. We talked about autosome versus sex chromosome. Our sex chromosomes also have genes on them. And it turns out that the X chromosome compared to the Y chromosome is providing a lot more contribution. I don't think that's a surprise to any high school girls in terms of, oh, hey, the male chromosome maybe not participating that much. Human females, in terms of simply the sex chromosomes, XX, and human males, XY, traits like color blindness, red-green color blindness, hemophilia, that idea of like having a difficulty to clot in your blood, those are actually X-linked or sex-linked, but it's usually the X chromosome, as I said, given just the real estate that's available, that are at the heart of how those traits are inherited. So in males with only one X chromosome, you can't mask it. So if it's a recessive allele, you still are presenting it because you don't have a dominant allele to mask it. So that's another fashion of inheritance that you want to be aware of. If you have questions on that, again, please let me know. And then We also have non-Mendelian inheritance, because the sex-linked and X-linked traits, either term is fine, it just tends to be X. Those still go by the Punnett squares that we were just doing. But non-Mendelian inheritance is where all of a sudden these laws of dominance or segregation or independent assortment don't hold. And incomplete dominance and codominance are examples of that. Classic example would be red roses and white roses. If all of a sudden you got out pink roses, it doesn't sound like the red masked the white, they mixed together, and there was an incomplete dominance. So a blending of the traits, something in the middle. Codominance would be red with white spots or white with red spots. Both traits are being expressed. So those both break the law of dominance. Polygenic inheritance is really just this idea that certain traits have a ton of genes that are involved in order to make those be expressed. We often talk about height in pea plants. I did it myself today, but that's actually a very complicated thing. There's every possible height available, right? It's not like you're just at quantized of this inch or this inch or this height. So that's an example of something that's polygenic, more genes involved in that. And I think, again, the big things here would be meiosis and the inputs and outputs, and then Mendelian genetics and Punnett squares, inputs and outputs, and what those genotypes and phenotypes mean. I mentioned briefly that environment, we would discuss its role in this. You can have a particular genotype, identical twins can occur, obviously, and then they themselves are still different people. The environment impacts them differently. Even if we're not talking about identical twins, we're just talking about people that may have the same gene, be it through relation or just, very little variation in that gene. They could still express that differently because of their environments. Anything from medication to diet to physical activity to countless different things. Other genes that are in their genome and being expressed. And then the other thing you may come across would be rules about a test cross, which is really just the idea that Mendel had his true breeding plants, and he kept wondering, okay, how come all of a sudden I'm getting a short plant when I'm breeding these two talls together? Well, it's because when you only have true breeding plants, that's essentially like with your recessive trait, that's a test cross, because every single time you won't express it. If it's just providing recessive alleles, and then it's essentially, if you get the recessive trait, then the other parent must have had the recessive allele as well. And then something else that can break one of his laws, linked genes. These are not going to follow the law of independent assortment. These genes may be so close together that they're always together. In that meiosis process, in that meiotic event where the individual chromosomes are going to different gametes, if genes are super close together, you're probably not going to have a crossover event. They're probably not going to recombine with something else because there's no space for that to actually happen physically. So the linkage of genes is based on their distance from each other. And then we can determine how often they recombine, and that's a recombination frequency. All of this, just additional vocabulary to get you comfortable, but at the same time, big points, linked genes are linked and they get inherited together. Okay, I think we are on to our question. So a couple of bits of news about these questions. College Board has gone ahead and indicated that the type of free response question you will have will be 1 and 4. There's going to be the longer one, 25 minutes long. It's a question 1. Set of data, you analyze it, you're going to be graphing something, you're going to describe something, explain something. You have a bit more time on those that are longer, but pay attention to those when you are looking at free response questions in your prep. Question number 1 in each of the years that are available. And then question 4, the shorter one. This is going to be a 15-minute question as opposed to the 25, which means a little bit longer than a typical number 4, perhaps, but much more just describe, identify, something like that, a little more manageable for us to do together in class today. So I found question number 4 from the year 2015 was about mitosis and meiosis. Here's an example of something that you need to know and express on this topic. And we are going to go over mitosis more, but let's see if we have enough. Both mitosis and meiosis are forms of cell division that produce daughter cells containing genetic information from the parent cell, which you all already know now. Describe 2 events that are common to both mitosis and meiosis that ensure the resulting daughter cells inherit the appropriate number of chromosomes. So it's a bit more of a functional aspect, and it's just description. And then the genetic composition of daughter cells produced by mitosis differs from that of the daughter cells produced by meiosis. Yes, we talked about that as well. Describe 2 features of the cell division processes that lead to these differences. Describe 2 features of the processes that lead to these differences. We also highlighted those. So I'm going to give you a little bit of time to think about that. Not as much as I would love to give you, but you can always pause this, continue to work, and then see the scoring guidelines. I'm going to be bringing those up very shortly. In the meantime, if you have any additional questions, let me know. Okay, so what do we need to get our full points on this question? Hello again. That is because I am now sharing this. Okay, so describe 2 events that are common to both mitosis and meiosis that ensure the resulting daughter cells inherit the appropriate number of chromosomes. There are many options here. You can only get 2 points. So that would be why I'm doing the larger cursory review. That didn't get into a lot of the spindle elements. I will get into that when we come back to mitosis, but the microtubules attaching to the chromosomes, they do that in a fashion such that they get pulled to offer up the correct number of chromosomes on both sides of the cell. The way in which the chromatin condenses, that's actually in order to organize everything, right? We're not bothering to express anything. We talked about feeds on a string last week. We're not trying to express anything. We're just compacting down the U chromatin that's being expressed, wrapping it around proteins and forming the nucleosomes, and then wrapping that up and supercoiling that until we have condensed chromatin, condensed into chromosomes. Their alignment, how they do that on the metaphase plate, I talked about that. Their separation, now instead of the spindles doing their job, we got to make sure the centromeres and the chromosomes also do their job. They're offering up separate points for each of that. So if you really wanted to focus on that, you could. We'll talk more about the checkpoints in the interphase and when we go over the cell cycle in more depth. And then replication or synthesis of the DNA preceding the division event, and then the cytokinesis separating. It's really just go through logically, prophase, metaphase, anaphase, telophase, what could happen to make sure that we're getting the right number of chromosomes on each side. It's not too deep. It's just what you would think logically is the case through the observations we made. Okay, and then the second part, describe 2 features of the cell division processes that lead to these differences in their genetic composition. In mitosis, what they're doing here for the points, you get 1 point for each row and 2 points maximum. So that means that you can actually indicate just information about the number of divisions and the resulting cells. That would inherently indicate that mitosis and meiosis are different, because we end up with 4 gametes in meiosis, but only 2 daughter cells in mitosis. So there we already see how, yes, we started with 1 parent in both cases, but there's 2 divisions in meiosis. That's another way in which you can separate these 2. That inherently means different genetic composition. The ploidy, oh, look at that. They use a word that I like as well. So everything is identical in mitosis. But in meiosis, you have half from one parent, half from another. It doesn't mean you are all of these chromosomes from mom and all of these chromosomes from dad. But however it works out, you have 1 copy of each of the chromosomes, and 1 could be from mom or dad. The separation of the chromatids crossing over as the recombination event, or the way in which the chromosomes, they themselves, separate in mitosis versus meiosis. This does not happen in mitosis because you only have replicated DNA and you're making these identical cells. But in meiosis, we talked about the two different divisions and how first you had the crossing over, but then you also had the way, well, whose copy of that chromosome am I getting, mom or dad? And that could be different for each chromosome. And then how those ended up separating to form gametes is another layer of diversity. So I hope that this helps clarify the big picture stuff for genetics and inheritance. If you have any questions, please let us know. Hashtag PMTV, @mentalmarkets, email classes at prepmatters.com. And then next week, we are going to have our exploration of the cell. At this point, we've talked about everything going on, all the way up from molecules to how information is being passed from cell to cell. Now we got to get to what is this cell actually doing? Why does this matter? Why is this the foundational unit of biology? It starts really getting good. So I hope to see you then, and good luck with your prep.